Well, good evening, everybody. My name is Lou Siegel. I'm with the New York State Marine Education Association, and I'm hosting this meeting for Lisa, our president, who's present with us but not able to host the meeting. She's present with us from a hospital bed, so that's a different discussion for another day. At any rate, we're really honored today, and I'm very excited about having Dr. Ellen Pikitch as our speaker this evening, our first Thursday speaker. I think it's about a year now that we've been running these first Thursday speakers, and each one of them has been wonderful in their own right. And again, I'm very happy to have Ellen speaking tonight. Dr. Ellen Pikitch is currently the inaugural endowed professor of ocean conservation science at Stony Brook University and the champion of the Shinnecock Bay Hope Spot. Sylvia Earle and Mission Blue designated Shinnecock Bay as a Hope Spot in 2022, the first Hope Spot in the state of New York. Ellen's life work has revolved around unraveling the mysteries of the ocean, advocating for its protection, and inspiring generations of scientists and conservationists to safeguard our planet's most vital resource. Her work has truly transformed the way the ocean is perceived and managed, and she is frequently called upon to provide her expertise at professional meetings, global scale fora, and the oceans, and to decision makers at the state, national, international, and global levels. Currently, Ellen's areas of focus include marine protected areas and the use of eDNA technology. A native New Yorker, she has traveled to more than 110 countries, has authored more than 200 scientific and popular articles, and edited 2 books, including this one, which I have with me. Dr. Pikitch will describe the 10-year journey to restore and understand Shinnecock Bay, which revealed many secrets. And again, Shinnecock Bay is near to the people in NYMSEA because over the last 40 years or so, we've had several conferences at Shinnecock, at the college in Shinnecock Bay. And the Marine Science Station at the college in Southampton is a magnificent facility that if you've never visited, you definitely should take a trip there. At any rate, I'm going to turn it over to Ellen now, and I'm sure we're in for an exciting talk. Okay, Ellen, take it away.
Okay, I'm ready. Thank you. It's a great pleasure for me to be speaking with you all tonight. And Lou, it's great to have you as the host. Lou, as many of you on this call know, was my high school marine biology teacher, and he launched me on this amazing career that I've had as a marine biologist and turned marine conservationist. And without him, who knows what I would have ended up doing. So I owe a lot of anything good that's happened in my career to Lou Siegel. It's also great to see so many older classmates. We're all older now, I guess. But you know, it's amazing how many of you are still engaged in this area, and it's great to see you again. Okay, so I'm talking to you from my home. I have a house out in East Quogue, right next to Shinnecock Bay, and one of the reasons that attracted me to coming here was when I came to Stony Brook University. There was a marine station that Stony Brook had just taken over. It had gone out of business a couple of times, and Stony Brook took it over, and it was going to be 7 miles away from my house. So that sounded really attractive. And as Lou said, we've got a state-of-the-art marine lab out here, a fleet of research vessels. And one of the things I tried to do right away was to see what could I do in my own backyard. And I'm going to talk about what I've been doing in Shinnecock Bay tonight. So as many of you know, Shinnecock Bay is a bay on the south shore of Long Island. It's got 9,000 acres of open water, salt marshes. It's a regionally significant habitat for fish and shellfish. It's a really shallow water bay. The water spends a long time here, and it's highly vulnerable to eutrophication. Another thing I'll point out while I have this up is, if you can see the little arrow going through, Shinnecock Bay has an inlet, the Shinnecock Inlet, to the Atlantic Ocean. And that's an important part of the story for a lot of reasons. The fact that there's exchange with the Atlantic Ocean water means that this eastern part of the bay has typically had better water quality, and along with that, a healthier ecosystem than the western part of Shinnecock Bay. So as I said, when I came out here, I started at Stony Brook in 2008, and I found out from my colleagues who'd been working here a lot longer than I'd been here, that some really terrible things were happening in Shinnecock Bay. This is what the water looked like a lot of the time. This is an example of what brown tide looks like. I think of it as a spilled cup of coffee. And it was around pretty much every single year. And during those first few years I was here, the duration of those brown tides was increasing. There were also red tides, rust tides, and just like all plants need sunlight to grow, so do seagrasses. And with the brown tides came the loss of seagrasses. That was one of the main factors, because the water was so brown and sunlight couldn't penetrate through, that the seagrasses could not survive. The water quality was terrible. The clam population collapsed, and it was a really degraded ecosystem. And I thought, wow, what am I getting myself into here? When the red tides started to happen, which was actually only just a few years after I got here, I got really worried, because red tides can cause paralytic shellfish poisoning, which can be fatal to humans, and if not fatal, can cause nerve damage. And for a series of years, about half of Shinnecock Bay had to be closed for shellfish fishing for a good portion of the spring, because of this threat of paralytic shellfish poisoning. So this isn't just, something that affects fish, which are very important to me, of course, but not everyone, but actually people as well, human health. What happened with the seagrasses in Shinnecock Bay was something that was happening throughout New York State during the period since 1930. Between 1930 and 2009, it was estimated that there was a 90% loss of seagrasses in New York State. And again, that was caused directly because of these harmful algal blooms that were due to increased nitrogen loading. And at the time of this report, this figure from the report, they had estimated that by the year 2030, seagrasses would be extinct in the state of New York. What happened to all the shellfish, the clams, the oysters, the scallops? Well, that was due to a whole host of things. One of them was plain old over-harvesting. The rules for fishing weren't tough enough, and too many shellfish got taken out. There was habitat destruction and poor water quality, and all of those together led to a situation whereas New York, the South Shore of Long Island, used to supply about 2/3 of all of the bivalves eaten in the country, it went into a precipitous decline. So in learning all of this and speaking with my colleagues, we said, we shouldn't just be documenting the problem. We should try to fix this problem. And so we decided, and it was a risky proposition, I have to say, that we would try to get support for what we call the Shinnecock Bay Restoration Program. And we were able to obtain a sizable amount of funding by the year 2012. And that's when this restoration program was launched. The heart of the restoration program is what we call hard clam sanctuaries. Hard clams, that's easy to understand, but they're called sanctuaries because they are no-take areas, analogous to marine protected areas. And the local authorities were happy to help us get that no-take designation and to make sure that the area was patrolled so that it was actually abided by. We planted adult clams, large adult clams, for a bunch of reasons. One is that we didn't have to worry about, if you plant tiny seed clams, as they're called, the predators that are in the system can eat them, and then you can end up with no clams. So we got clams that were large enough that no predators in the bay would eat them. They would already be a reproductive condition. We had to actually buy them on our grant, and they were expensive. We estimated that we needed many millions, 33 million bivalves, in order to bring the water quality to a level that would potentially eliminate the harmful algal blooms. And there was no way that we could afford to buy 33 million clams. So we had to hope that by bringing in adult clams that were reproductive size and age, they would be successful, they would come into reproductive condition, they would spawn and make lots of babies, and those babies would make babies. And an interesting thing is that hard clams can live for about 40 years. So once you get an adult established, it can be there for a really, really long time. And the idea was that natural reproduction would then take over and expand the population. And why did we want hard clams? Because clams and other bivalves would help to restore the natural filtration capacity of the bay. They clean the water. And we hope that by doing this, restoration would follow. People said, we were nuts to think that this would work. And we ourselves wondered about it. Would it work? Would it not work? Would it be enough? And, here we are more than a decade later. But other solutions like building sewage treatment plants and changing the whole infrastructure that doesn't exist out here, those are things that we knew would take a really long time to get done if we could ever get the permission and the money to get it done. And things were so dire that it was important to do something that could help immediately. And so the idea of these hard clam sanctuaries seemed to be worth going after. Initially, we weren't able to get permission to build oyster reefs in Shinnecock Bay, but after a number of years, we were also able to do that, to get permits to build some oyster reefs. And now we have 7 oyster reefs in Shinnecock Bay. Oysters are highly resilient to degraded water and do a great job filtering the water. And then the seagrasses, the eelgrass. We tried a number of approaches, and what we found worked best for us was to actually take the seeds and to spread them to new areas. Reseeding eelgrass in the bay. We held a number of annual community events to get the community involved in this. And what happened? Well, what happened is that the clams did what we hoped that they'd do. In fact, they did it even better than we thought. They reproduced, they made baby clams. Those baby clams grew up and made more baby clams. And the hard clam sanctuaries themselves are very, very healthy. But in addition, the areas in the Eastern Bay that are still and were still open to clam fishing also received a huge amount of recruitment of clams. And the clam fishery today is back to a level where it was in about the 1970s. So there's been a huge increase. So it's been good not only for the environment, but it's been good for the economy, good for the clam fishermen. Here's what happened with the brown tides. that I'd look outside my window and see these horrible brown tides. Well, it looked like things were starting to change. And then 2016, there was a big spike. And then 2017, the brown tide cells per milliliter went down. And that was the end of the brown tides in Shinnecock Bay. So Shinnecock Bay went from looking something like this to what it looks like today. And we actually did turn brown tide blue. That's one of the slogans of the Shinnecock Bay Restoration Program. And now Shinnecock Bay stands as a beacon of hope, not just to us, not just to our community, not just to New York, but to a global audience, that we can make a difference. We can turn things around. If we use good science, if we're persistent, if we're adaptive, and we work hard, then here's an example of a place that was turned around. A lot of the science involved in the hard clam sanctuaries was published last year by our large team in the Journal Frontiers of Marine Science. This is open access, available to any of you for free. So I've talked about clams and I've talked about eelgrass and oysters, but what about the fish? What about the fish? Some of us really, really care about fish. And we didn't do anything, and this is common in restoration efforts, we didn't do anything directly to restore fish. Like we didn't have hatcheries and we didn't put baby fish in the water or anything like that. So what we had to hope was that this restoration would filter up to the broader ecosystem, and that by cleaning up the water, the habitat, by getting more seagrass, more eelgrass, which is critical habitat for young fish, that we would actually start to also see improvements in the fish community. And in order to do that, we had to monitor the fish community. So here's some of the team. We've had lots of people work with us over the years, lots of students who've gone on to other careers, and it's been a great program. So our main method of assessment has been bottom trawling. That's the method that had been used here. It's still used in a lot of parts of the world as an assessment tool. Of course, we use a very small trawl net, and we don't put it out for very long. We actually only have to have the trawl out on the bottom for about 3 minutes to get enough data to be able to compare catch per unit effort in different areas and in different years. So it's trawling, but it is low impact trawling, relatively speaking. And we now have the longest standardized trawl survey data set in Shinnecock Bay, and it's pretty comprehensive. So this map shows where some of the trawling sites have been. We had to make some changes because some of the initial sites were in areas that we later turned into hard clam sanctuaries. And we didn't want to go trawling and trawl up all the clams that we planted. So we had to make some changes in that. So let's take a look. What did this trawl data series tell us? One thing that I was really interested in looking at were the regional differences in the different parts of the bay. So the Eastern Bay in kind of a greenish color here, as I said, it always really has had better water quality because of exchange with the Atlantic Ocean. And the Western Bay in a darker blue had always the poorer water quality. One of the ways that we thought we might see changes due to the restoration effort would be if the fish community in the Western Bay would start over time to look more and more like the fish community in the Eastern Bay. So that was one of our metrics. Let's look at, this is actually a pretty recent, this is from 2022, pretty recent pie chart of fish species diversity, and the Eastern Bay species diversity is on the left. You can see that there are more species in the east, and the distribution of individuals among species is much more uniform in the east than in the west. In the Western Bay on the right, it tends to be dominated by, one or two or three species. So a lot less what we call evenness in the biodiversity and fewer species overall. So the fish community still does reflect a difference in the water quality, but this is actually a heck of a lot better than it was when we started out. With invertebrates, similar trend, although with invertebrates in general, there are fewer species in Shinnecock Bay than there are fish species. The Western Bay is heavily dominated by blue crabs and grass shrimp, two species. And the same two species dominate in the east, but there are more other species. Okay, so if we look at the catch per unit effort in the Eastern Bay minus the Western Bay, then we get the graph that we see here. And what you can see is a trend as you go from left to right of that difference is getting smaller and is changing. And in fact, near the end of the time series, the Western Bay is actually a little bit above the Eastern Bay in terms of the catch per unit effort of different species. And some of these trends are significant, statistically significant for some species. Atlantic silversides, one of the forage species, that's a highly significant change, an increase. Weak fish, a decrease. In the invertebrate community, we found significant changes for spider crabs, green crabs, and mantis shrimp using this technique. In terms of diversity, there haven't been big changes in either the eastern or western portion of the bay for either fish or invertebrates over time, but there is a huge difference in the number of fish species, much greater than the number of invertebrate species. Okay, another way of looking at, is the restoration making a difference for fish? This might be a little bit easier to understand than the last graph. Is the catch per unit effort changing, and is it in particular increasing over time as the restoration progressed from 2012 to the present day? And here you see what looks like and actually is a statistically significant increase in the catch per unit effort, which is related to the relative abundance of fish over this time period. The invertebrates do not show a significant trend. This is giving more detail on specifically what's going on with these fish. And what you can see are the total fish population is in this purplish color. Within that, there are bars, and the bars represent the forage species. Forage species can feed on plankton. A lot of bigger fish cannot feed on plankton. So these are low trophic level species that feed at the foundation of the food web. And they're called forage fish because everything else pretty much forages on them. Atlantic silversides and menhaden showed statistically significant increases over the period of time of our study. And that's what we would expect. If anything is going to show a change first, it's going to be the forage fish. It's going to be the fish that are most impacted by improved water quality conditions and the elimination of harmful algal blooms. This is showing more species analyzed in the same way. And interestingly, what I was happy to see is that we actually also saw a statistically significant increase in horseshoe crabs in Shinnecock Bay during this time period. They're always a group of species that we worry about. Green crabs, which is known as it's among the 100 worst invasive species in the world, actually showed a significant decline, which is good news because they can be very destructive. So as I said, we started out and have continued doing bottom trawling as one of our survey methodologies. But a few years ago, we started using something called eDNA. And the E in eDNA stands for environment. And in this case, the environment is the water. From a sample of water, there are sloughed off skin cells, there are secretions, scales fall off a fish. There are fragments of DNA from the fish community in that water. And we now know that if we take a water sample carefully and analyze it and look at the DNA, it can tell us a huge amount about the fish community. So it's a powerful tool. It's a relatively new tool for advancing ocean observation, particularly in the marine environment. I was lucky to publish this article in Science in the year 2018, which was a perspective piece on eDNA. And at the time, so what it enabled me to do is to take, basically review the literature and to make some predictions about what would happen in the future and talk about what I found. And what I found was that eDNA outperforms other assessment methods. It's non-invasive, non-destructive, neither to species or habitat. It's more powerful at detecting species, and in particular, it's been used to detect species that are relatively uncommon, rare species. It can also sample the full size spectrum of life, anything from, the smallest cell to a whale shark or a blue whale. Because, imagine a little research trawl is not going to catch a whale or a whale shark. It's not going to catch fast swimming fish. And so when you use a trawl and use other methods, they are size selective, and we miss out on the very big things and the very little things. Whereas eDNA has the capacity to look at the full size spectrum of life. We can use eDNA almost anywhere because all we're doing is taking water samples. So we can use it in highly sensitive habitats. It's a lot less labor intensive, and it leaves a much smaller carbon footprint than bottom trawling or other assessment methods. So I got so excited writing this piece that I said, hey, I think I'll try to use this in Shinnecock Bay. And so I set out to learn how to sample for and use eDNA. And this is what we've been doing is pretty low tech. You can see that we have a pole, and at the end of the pole, a bottle that's been sterilized beforehand, sanitized. We rinse it in situ. We take the water sample, we close it up, put it on ice, rush it back to the laboratory because it has to be analyzed the water has to be filtered within 24 hours, and then the DNA extracted from the filter within another 24 hours in order to get good data. And here are all the different steps that need to be taken. It's a complicated process, but in the end, we get from what's called bioinformatic analysis, we get the DNA sequences of the species that were there, so we know what species were there, and we can get sequence counts, which can tell us something about how abundant those species are. I know this looks complicated, but bear with me for just a minute. So this shows all the species that we've seen either in trawling or with eDNA. And everything in blue is from a trawl. Everything in the reddish, orange, yellow category is from eDNA. And if you focus on this left-hand part of the graph, these are the species that we call the regulars. These are the species. There's a lot of variability in a system like Shinnecock Bay. So there are some very rare species that we don't see very often. But there are a group of regular species that we've seen every single year since we've been doing the study. And this graph was made when we had 11 years of trawling data. So these species that are in this box are the regular species in the trawl. And we were happy to see that the 3 years that we did eDNA, those same regular species showed up in every year of the eDNA sampling as well. So eDNA was able to detect not just rare species, but the common regular species. And if you go out and look at the right-hand tail of this graph, you see more red and yellow and orange than you do blue. Those are species that we found only once or twice. And what this is telling us is that eDNA is better at picking up those very rare species than, because there's more red, yellow, and orange than there is blue. So it's better at detecting rare species, and it's at least as good at detecting common species as trawling. This was pretty interesting. We decided to focus on elasmobranchs one year, and that's a group of species that we don't see very often in trawling. So in all the years that we had been studying trawling, we had only found 3 species in the trawls. And when we started doing eDNA, we found 4 additional species uniquely found through eDNA: sand tiger sharks, short-fin makos, cow-nosed rays, and little skates. So in just 1 year of sampling, we increased by 133% the number of elasmobranch species that we knew inhabited Shinnecock Bay. So sometimes, with this kind of technique, a little bit of study, a little bit of sampling can yield pretty amazing results. We also found that 96% of all of our water samples contained some elasmobranch eDNA, and 100% of the elasmobranch samples contain smooth dogfish. So we have seen smooth dogfish in our trawls, but not often, and certainly not to the extent where we would think that they would be everywhere in Shinnecock Bay. We also found that there were 7 species of fish that we found only in the newly created restoration areas. So that was good news. It does say that the restoration is not only increasing the abundance of fish, but it's bringing in some new species. And here they are. So these are species found only in restoration sites: Atlantic moonfish, California anchovy, cow-nosed rays, sargassum triggerfish, long-nosed butterfly fish. A lot of people are amazed that we get tropical species in Shinnecock Bay, but we do, especially in the late summer and fall, when the Gulf Stream changes the current patterns and those tropicals are brought up. Lookdowns and sea robins. So what have we learned? Well, we've learned a whole bunch of things. We've basically confirmed that eDNA is able to detect a very broad spectrum of life. We want to, in the future, extend the eDNA research to an even broader spectrum of wildlife, including marine mammals, sea turtles, and seabirds, to gain a whole ecosystem perspective. We have shown, and it makes sense, that eDNA can be used in very sensitive habitats where other methodologies can't be employed, and that we found 7 new species in those sites. We also learned that we had been misidentifying some of the species through visual identification, even though we have keys on board and we do try as hard as we can to correctly identify species. But when we started getting the DNA sequences, we realized that some of the species we've been calling one thing were actually a different species. So another advantage of DNA is that you don't have to be able to visually determine the species identification. And eDNA sampling is more cost-effective, less labor-intensive, and has a smaller carbon footprint than trawling. So what's kind of the next phase? What have we been doing in terms of eDNA? Well, now in Shinnecock Bay, we have a very rich long-term database, and we've got access in Shinnecock Bay both to the ocean and to estuarine habitats. We've got lots of people and facilities. We are expanding our partnerships. We're working more closely with the Shinnecock Indian Nation now, and we are expanding our partnerships with other organizations that are interested in studying or doing community outreach on Shinnecock Bay. And we think it's an ideal test bed because of this rich database for developing and testing new and emerging technologies. So just this last summer, we had Schmidt Marine Technology Partners brought out 4 new technologies. One of them was an eDNA sensor that can actually do the eDNA analysis in situ in the water. We had some autonomous vehicles that could be set on automatic pilot and go around the bay and measure water quality, and so on and so forth. And what we're hoping to do is to continue with this testing of these emerging technologies and providing proof of concept so that they can be deployed more widely in all the ocean regions of the world. So this last question is, can we really conclude that restoration of hard clams, eelgrass, and oysters actually improved the fish and invertebrate communities of Shinnecock Bay? I think the evidence is pretty strong now. We showed that fish abundance has grown with our trawling data. And in fact, it's grown between 3.5 and 4-fold from 2012 to 2022, big increase. East versus west, the catch per unit effort of species in the 2 different parts of the bay is now more similar. And I didn't show this graph, but we have also found that the fish CPUE significantly increased in Western Shinnecock Bay, even more than it did in the eastern part of the bay. So the part that was more degraded has shown the biggest increase in fish abundance. The 7 species we saw in our restoration site says something to us about the importance of restoration in species diversity and abundance. And the forage fish CPUE quadrupled. And again, we expect forage fish to be the first to respond to improvements in water quality and in ecosystem health. So when will the higher trophic level organisms follow the forage fish? We knew that there would be a lag time, and we're hoping that we'll see more and more of that happening in the years that come. So after about 10 years of doing this work, it was very clear to us that our restoration was successful in a whole host of ways. The brown tides were eliminated, the clam population was flourishing, seagrasses made a major comeback, fish and wildlife were thriving, and no more red tides. People can enjoy a healthy and safe environment. And so we at Stony Brook got involved with the Explorers Club, headquartered in New York City, and together we nominated Shinnecock Bay to be considered to be a hope spot by Mission Blue. And as Lou gave the punchline away at the beginning of my talk, in 2022, Mission Blue actually did go ahead and recognize Shinnecock Bay as the first hope spot in New York. And it's one of about 150 hope spots all around the globe. It puts it in a league that is in the same league as places, iconic ecosystems like the Galapagos, the Ross Sea in Antarctica, the Sargasso Sea. These are famous places and, known to pretty much anyone. And now Shinnecock Bay is up there in the big leagues, and it's getting a lot more attention. And the lesson that it tells us about what's possible and what we can do, and that there are reasons to really hope, not just hope, but do, and make a change the world. These lessons are being spread pretty widely. So, the examples go from the hyper-local, like the Southampton Press, to global. I spoke at the UN at a special meeting between scientists and diplomats this June, and this was picked up, among other places, by National Public Radio. And the diplomats at the meeting said that the Shinnecock Bay is a model for ocean conservation globally. We've also had some TV coverage, including a pretty detailed, comprehensive series where the Shinnecock Bay Global Hope Spot was featured by NBC News. And then this year, Sylvia Earle joined us for the day. We have Richard Garriott standing to her right, who's the president of the Explorers Club. I'm to her left. We have my colleague, Chris Gobler. We have one of the Baymen and Southampton trustees, Ed Warner. We have a member of the Shinnecock Indian Nation. We have technologists here, and we had the official ribbon cutting of the Hope Spot in June. So as I wrap up, what do we hope to do as for the Hope Spot going forward? We're going to continue to monitor the bay's health. We have new ideas for additional restoration. This idea of Shinnecock Bay being a test bed for new science and technological innovation is something that is gaining more steam. We're enhancing our engagement with stakeholders, and we're spreading the word locally and globally. 2024 is going to be a big year. We're making plans. We've been making plans for a major expedition of Shinnecock Bay. Sometimes I call this the All Shinnecock expedition, because it's not just going to be looking at the fish and the invertebrates and the clams, but at seabirds and marine mammals. And also, we're going to be working directly with the Shinnecock Indian Nation and other organizations that we're in conversation with now that study other species groups in the bay. And we're going to cover all waters, both in the land and the sky of Shinnecock Bay, and use new and emerging technologies. So many, many thanks to the team. These are my main co-conspirators in the Shinnecock Bay Restoration Program, playing with clams. This is actually how you plant clams. You just throw them in the water and they find their own way down. And there are lots and lots of people to thank, without whom this project would never have been possible and would not be possible. And I've got a lot of them here, not all of them. And that's what I have to say. So I'll open it up to questions, and
Thank you.
Stop sharing the screen.
Thank you so much. It really is a great story. The most out of all of it, the most interesting thing I found was that you used the older clams, the cherrystone clams, as opposed to the smaller little necks, which further to the west, they planted mostly the smaller clams, the little necks. I don't think they've been as successful as you've been. That was I hadn't heard that before, that you use the older clams like that.
Yeah, well, actually, so, we don't always tell the whole story. Before we started the Shinnecock Bay Restoration Program, before we got big money for it, we were donated seed clams, and we used seed clams early on, before 2012.
Right.
And we were very excited about it because other people were using that. We got them for free. We put them in the water. And then we went down, we went down diving to look for them, and they were all gone. They didn't survive. They were all eaten. And, that was a real wake-up call. And it said to us, we just can't use these sea clams. We've got to use something bigger. Yeah. And so by the time we got, we were able to get more significant funding and could purchase adult clams, we knew that was the way to go.
Yeah, so, no, that's a great strategy that I hadn't heard before.
Yeah, it works. It works if you can get them, large enough to begin with, then they're going to stay with you, and they're going to be, again, ready to reproduce right away. So the increase in the population could begin. And
if I can, just one more thing. The eelgrass, have you seen significant growth of I don't think you showed a slide on that. Have you seen significant growth of the eelgrass?
We have. So Brad Peterson is our eelgrass specialist, and I didn't have any. I should have asked him for some of his slides before I put this together. But the amount of eelgrass cover has increased by more than fourfold over this time period. So it has been significant.
Great. All right, anybody else have questions?
I have a question. Thanks, Ellen. Really great. The power of eDNA is impressive.
Oh, mommy.
And I know that Howard Rosenbaum was doing stuff in New York Harbor looking at eDNA. I'm just wondering how you can, let's say you've got some rare species that you're picking up that DNA from. How are you sure that they are actually in the bay, or the eDNA hasn't been sort of flushed in when it comes in through the inlet, or is getting there through the fecal matter of some predator that's like a harbor seal or a gray seal hanging out in the bay?
That's a really good question. One of the great things about eDNA in aquatic environments is that it degrades really quickly.
Oh, there you go.
It tells you the here and now.
Oh,
It tells you what is here right now, not what was here before or what was here far away.
Mhm.
It doesn't persist very long in the water. It's the degradation rate varies with temperature. So the warmer the water, the faster it degrades.
Sure.
So it's a great way of getting a snapshot of what's here right now.
That's great. Thank you.
Sure.
That was my only question. It's nice to see. I've been doing stuff for the seals over at Shinnecock Bay for a while now with my students. And we take the public out to see them. And some of the seals in Shinnecock Bay I also see over at Cubsog Beach. At least a couple of them that I can ID and see there in both places pretty regularly. So it's nice to be able to perhaps do more stuff there.
Yeah, so I am really interested in looking at the seals, and especially we try to work a lot of the year, but we don't do as much work in the winter. And seals are out there in the largest numbers.
Yeah, right. I'm out there in the winter with them sometimes. So
yeah,
happy to work with you somehow.
That'd be great. Who's saying you go next? Is that you, Lou? Should I pick? I saw Joel has his hand up.
Yes, it's a great presentation. Great presentation. I appreciate it. I learned a lot.
Thank you.
I basically have two questions. What sort of funding has your project received from the federal government? And also, with such animosity existing today and denial of climate change and getting funding for projects like yours, how are you able to overcome that?
So most of the funding for this project has come from private foundations. Very little has come from the federal government. And the interesting thing to me was that, a lot of times people from different walks of life don't see eye to eye on things. And I did worry, how are we going to get people behind the concept of working on the bay, especially things like creating hard clam sanctuaries, where we were going to say, you can't go fishing there anymore. But it was so obvious to everyone who, whether they were commercial fishermen, recreational fishermen, people who just enjoyed recreating on the bay, that things were so bad that something had to be done. It was just obvious. The fishermen who were fishing in traps, they would go and get their traps and see that the tides, the brown tides and red tides, would sometimes leave the fish that got caught in the traps would be dead. And they were seeing fewer and fewer clams. The fishery was had gone downhill. The red tides, the threat of human health impacts, paralytic shellfish poisoning, that was scary to everybody. So I think, sometimes when you know that there's a common enemy and that common enemy is a horrible environment that needs fixing, people do rally together and come together to make a difference. And that's what happened here.
Thank you.
Thanks. Okay, Lisa?
Can you hear me?
Yeah.
Yes.
Okay.
What a trooper, asking questions from her hospital bed.
Thank you. Yeah, I wanted to hear you talk. I think the work you're doing is fascinating and of course very much needed. And the two photos you showed between the before and after, or of the before and after, are so striking of Shinnecock Bay. So you're doing amazing work out there. I like the partnerships that you are going to invest in, more, like the Native American nation out there, the Shinnecock Nation. I recently listened to a Zoom talk given by their director, and I think it's like one of 6 women divers diving for the sugar kelp.
Mhm.
And it's just fascinating what's going on out there, and it's not let's say as known as other sugar kelp aquaculture areas, off the coast of Massachusetts, let's say, for example, or
Mhm.
Maine. There's some pretty famous people. Bryn, I think his name is Bryn,
Mhm.
and so he has his company, and he's more advanced than these women. But anyway, back to you. I like the partnerships that you'll be forming or will be investing more in
Mhm.
as time goes by. And also I was wondering if well, here's my pitch. If NYMSEA could possibly do some kind of field trip out to Stony Brook, and maybe also combine it to see the women, aquacultural divers, and perhaps make that a destination, which I hope to talk to you about more.
Yeah, I would definitely be happy to arrange that. We've done field trips ranging from, a few hours out on the boat to days. One of the things that we've done, we actually work at the Master Teachers Program of the state of New York. And so we've had workshops that last several days. The master teachers come, they get to be involved in every aspect of the research that we do. And they get to know one another, and a lot of them come back and do great things to help us. So
that's great.
yeah,
Maybe
so
that's something we could set up with Chris Paparo and you together.
yeah, Chris Paparo is a great resource for my group and for the whole project. A lot of you may not know him, but he's a facilities manager, and he's also a fantastic naturalist. He knows all the species. If we're unsure of a species, we usually text him a picture and say, "What do you think, Chris?"
Yeah, no, he's been great. He's a member. He's been great with this.
Oh, he is?
He presents
Okay.
at most of our conferences.
I've known him for 30 years.
And actually, he was the first Thursday speaker 2 months ago.
Oh, okay, great. Yeah, so on a lot of these trips, he's usually out on the boat with us.
That would be great. I think teachers would love to participate in the different phases or facets of your research, or how the research is done, in some kind of workshop. But whatever comes out of it, just talking about it is also pretty fascinating. So thank you. I'll contact you
Again,
later.
my pleasure. Also, if any of you or any of the members are local to Shinnecock Bay and want to volunteer, we do always have a need for volunteers. So when we go out trawling or go out eDNA sampling, or people who can operate small boats, for example, you have to be vetted though by the station. We're always in need of helping hands, and people who are dependable and reliable and enjoy doing it, and are fun to be with. So yeah, if we didn't have volunteers, it would be really impossible for us to get all this work done.
Oh, that's great. And also, you just reminded me, now that you are a new member, I saw.
Yeah, I actually finally joined. I'm sorry it took so long, but now I'm a member. I paid my dues.
Well, welcome, new member. And what I want to say is, there's a little bit of little story about our newly purchased ROV. And so you might want to read up on that in our current newsletter.
Okay.
And I was just thinking about how an ROV might be an additional helper in this kind of research, since we actually have our own equipment now. So keep that in mind.
Yeah.
We recently purchased a
Uh-huh.
professional level, research level ROV, and we're just testing it out now. But that would be a great way to see the bottom, directly as opposed to remote. Yeah.
One of the things, just to give an example of another need that we have. So the habitat, when we started the project, one of the things that we did was we mapped the habitat, and we did it the hard way, where we put out quadrats and we went diving, and we basically did it visually, and we had a pretty good habitat map. But now we know that that's completely out of date. Now there's seagrass where there wasn't seagrass. The habitats are all different. We need to update it, and we want to find a much more efficient way to do it. So maybe this ROV could help.
Absolutely.
That would be great. Just one more question, if I may.
Sure.
You said that you do the trawls for 3 minutes. That
Yeah.
seems like a really short time. Did
Yeah.
you come? You said that you didn't want to disturb the bottom. But
And we also didn't want to kill the fish.
right.
Bottom trawling not only destroys habitat, but if the
Is
fish
that like a
in-
standard thing to do it for such a short time?
it is for my study. It's not for a lot of other studies, but and I was worried, would 3 minutes be long enough to get enough data that would lead to statistically significant differences among, between areas and among years. And, I showed you that, yes, in fact, we have been able to get statistically significant information. So it's enough. And yeah, I actually I've studied bottom trawling. It's not my favorite method of either fishing or sampling. And I do, I know that the longer a fish is in a trawl, the more likely it is to die. And I used to think that they died, well, there are different reasons that they might die, but the main reason that fish die in trawls, if you don't know this, you can tell this by looking at video of them swimming. They try to keep pace with the trawl net, and they keep swimming and swimming and swimming and swimming. Eventually they get exhausted and they fall back into, this big group of fish in the back, and they're basically dying of exhaustion. So if you don't trawl for very long, you're not going to tax them very much, and you're going to have much healthier fish that survive. So we're trying to make, this bottom trawling as minimally destructive as possible. I think we're doing a pretty good job of that. And also the eDNA, my dream is to eventually possibly totally replace the trawling with eDNA technology. Now, there are disadvantages of that, in that there are some things you can only do by actually holding an animal in your hands and looking at, what it's eaten and things, and it takes some of the fun away too, to not actually see the actual organism. But I think that eDNA can do for us a lot of what bottom trawling has done. And for a lot of assessment surveys all around the world, it might be able to largely replace bottom trawling. So it's one of the things I'd like to see. I'm actually giving a talk at that. There's a World Fisheries Congress coming up in Seattle in March, and those happen once every 4 years. And I'm giving a talk on, can eDNA replace bottom trawling as the major survey assessment tool in fisheries today?
Great.
Wow. Congratulations on that.
Meryl, did you have a question, or
You have to unmute?
you have to unmute yourself, Meryl?
Okay. Fabulous presentation. Thank you. When you have a sample of the eDNA, genetic fingerprinting of various different species of fish, right?
Right.
So now we have something that is unknown and doesn't match with your
Meryl, you're coming in and out, Meryl.
Yeah, you're somehow your voice is
Try
okay,
and
you know,
talk right into the phone.
okay. Yeah, no,
It's coming through the car. So when you sample your eDNA, and I assume you compare it against an established library of DNA fingerprinting, right?
right.
For
Right.
some of the more, common ones. But when you have something that is not matching up with your library of DNA fingerprints, so to speak, you have
Mhm.
something that's unknown, how do you even begin to figure out what that unknown DNA fingerprint is?
Well, sometimes you just have to have patience and wait. Um, you may have heard, there's a lot of talk. One of the big controversies in marine conservation today is about deep sea mining. Should we do it or not? And people have argued, some people have said, oh, the deep sea, there's nothing there. It's void of life. And there are only these inert nodules down there full of minerals that we want to use for electric cars and other things.
Mhm.
So recently in something called the Calverton-Clipperton Zone, which is one of the areas that was moving ahead most swiftly for deep sea mining. Somehow some research organization got there and was able to sample using eDNA in the deep sea. And they found more than 5,000 species from a very small amount of sampling in this Calverton-Clipperton zone. And that has, I think, had a big influence on putting a halt to the movement forward for deep sea mining. Now, we don't know what those 5,000 species are. We don't know their names. We don't know for many of them what genus they're in, but we know that they have unique DNA sequences that have never been seen before. So we have that much information.
Huh.
Yeah.
Wow, fascinating. Thank you so much. You continue to make us so proud of you. Thank you.
It's amazing to have so many of you on this.
Yeah, it's really actually pretty funny.
Team.
Joel Terrett was also a teacher in one of the first teachers of marine science. And he actually codified the Regents Marine Science curriculum.
Oh,
So,
wow.
Yeah, actually, there's a copy of it on my webpage. So in around 1990 or so, he codified the whole, took a sabbatical, and he codified the whole marine science Regents curriculum at the time, which basically was 4 or 5 schools fighting with each other. Well, let's include this, let's include that, but Joel did this encyclopedic version of the curriculum that we were able to choose from and work from.
That's great.
The check's in the mail, Lou.
Right.
The check is in the mail.
The check for me or the check for you?
You.
Yeah. At any rate, Ellen, it was really great hearing you, and it's a great project. And having worked at the South Shore Estuary, I know what's going on and what went on in Shinnecock Bay. And again, NYMSEA from the 1980s has been going out to the Southampton campus for conferences, and they were some of our best times. Were out at the conferences and at the Marine Station before the new building was built, and going out on the Shinnecock and the Lucayo and the other boats, not the Lucayo, Shinnecock and some of the other boats that were there. And
the Peconic and the Pominock.
Right. The Lucayo shows my age. That was at CW Post. At any rate, thank you so much. And again, this recording will be on the website in a day or two, and hopefully this is just the beginning of a more involved relationship for NYMSEA and the Stony Brook campus in Southampton. So,
And you.
Thanks, Ellen. Great job.
Thank you again for inviting me in.
Thank you so much. Thank you.
Thank
And
you.
Lou, thank you again
Yeah.
for launching my career as my high school marine biology teacher.
You're lucky